And what are these two methods that Mendel used? One, we'll have the monohybrid inheritance, and two, we have the dihybrid inheritance. In monohybrid inheritance, here we have two different plants having a one control character. Like he used two different plants, a plant that is tall and one that is short. He tried to crossbreed these two plants to see what the offspring will be like. Will the offspring be tall, or will the offspring be what? Short, or will they be average? That is the reason why he's trying to use two different plants for a particular contraction what character, that is height. It can be complexion, fair and dark in complexion. How will the offspring look like? Will they be fair? Will they be dark in complexion, or will they be heterozygous in nature? So these are the characters that Mendel used, or the method that Mendel used in monohybrid. While in dihybrid was these are a kind of method used to contract two different plants having two different contraction characters. Contraction characters like you can take height and complexion at the same time. A height in the sense that we try to contract the height, tall and short, or like that's of the piston sativum that has yellow and green seed, wrinkled and round seed. You can crossbreed and see what it is. Filial generation will be to know the phenotypic ratio and the genotypic ratio. Okay, so let's look at one monohybrid inheritance. That the method that Mendel used. Like when he now realized that this piston sativum, that some are short and some we are tall. So he have to crossbreed these two plants. Assuming that this is TT stands for tall, while small t stands for short. So segregation must take place. We'll look at that. These two characters must segregate. That is tall and small tt. So in this case, the crossbreeders to this will give us capital T, small t. This to this will give us capital T, small t. This to this will give us capital T, small t. This to this will give us capital T, small t. This is the first filial generation in characters. Looking at this now, the offsprings are heterozygous in nature. We have capital T, which is the tall, and short t, this is how they are. So the phenotypic ratio here now is what? 4:0. Looking at this offspring here, this proves that all of them are tall because the dominant, the T there, the capital T there is the dominant gene. And all these offspring here, phenotypically, they will be what? Tall. So, in this case now, looking at these offspring now, they will be all tall. Not that the short trait is not present. It's there. But in subsequent generation, these short necks will do what?